What exactly is DNA? People blame it for everything from receding hairlines to Olympic-level metabolisms. It's spoken about like some ancient scroll written inside you, deciding your every trait before you've even had a say. But what is it, really? And why does this one molecule quietly run the show for everything alive on Earth? Tonight, we'll follow the story of DNA, from something you've vaguely heard about to something you deeply understand. You'll see how it explains why you're not just a blur of atoms floating about randomly, why your eyes, lungs, and fingernails know what to do and when to stop. You'll learn how DNA connects you not just to your parents, but to fungi, whales, and even bacteria. A bit awkward, yes, but scientifically useful. Everything alive, every blade of grass, every fly buzzing near your window, even that leftover mushroom in your fridge, is built using DNA instructions. The same molecule, doing the same sort of thing, in almost every living thing we've ever studied. It's astonishing in its simplicity, stubborn in its repetition, and utterly indifferent to your opinion of it. You might think of DNA as something rarefied or delicate, but it's incredibly common. Every cell in your body has it. You're sitting there, surrounded by DNA. You're basically full of the stuff. And yet, despite being packed with it from head to toe, most of us don't know how it actually works. A bit like car insurance. The instructions inside your DNA are not written in English, obviously. They use a code of just 4 chemical letters: A, T, C, and G. That's it, 4 letters. From those you get elephants, pineapples, tortoises, and teenagers trying to skateboard down staircases. Which, frankly, is a bit of a design flaw. But this isn't a story about human cleverness. It's not about some magic chemical that wants you to be tall, or smart, or fond of jam. It's about a natural process, one that has worked the same way for billions of years, a quiet system of copying, variation, and survival. No plan, no audience. Just chemistry, repeated until it accidentally got complicated enough to wonder where it came from. Tonight, we'll take our time. We'll explore what DNA is, how it works, how we found it, and what it tells us about ourselves, our past, and the universe we briefly occupy. It may not answer every question, but it will answer the right one. What is DNA, really? This leads us to consider how this molecule shapes your own life, starting with the things you've probably already noticed. We've named it DNA, but that only gets us so far. It's a tidy little abbreviation, short for deoxyribonucleic acid. It sounds technical, slightly intimidating, and vaguely important, like the sort of thing you'd rather not admit you're still confused about. So let's ask the actual question: what is DNA, really? Not what it does or what it's used for, but what it is. What is this molecule made of? Why does it matter so much? And how can something so small shape something as complicated and moody as a teenager? It's a question that lies at the heart of biology, and the answer, calmly and inconveniently, is both simple and impossibly layered. DNA is the molecule that holds instructions for how living things build and run themselves. It doesn't shout. It doesn't rush. It's just there, copied from one generation to the next, quietly organizing things. The shape of your ears, the structure of your bones, even your odds of liking coriander, all of it, shaped by this microscopic instruction manual you never agreed to read. And this isn't just a human story. DNA is shared across life on Earth. The same molecule that shapes your limbs also shapes the roots of trees, the fins of fish, and the eyes of owls. It's not a one-off invention, it's a system that works, and has kept working for over 3 billion years. Efficient, ruthless, and about as sentimental as a brick. To answer what DNA is, we'll have to look at chemistry, biology, history, and time itself. We'll explore how it works, how it copies itself, how it makes proteins, and how it sometimes gets it wrong. We'll see how people discovered it, slowly and stubbornly, often by mistake. And we'll learn what it tells us about who we are and what we're part of. Of course, there are complexities. DNA doesn't act alone. It works inside cells, surrounded by systems, signals, and constant molecular bickering. Some parts of it don't seem to do much at all. Others do things we're still trying to understand. And yet, somehow, the overall system mostly works. Which is arguably more impressive than it sounds. By the end of tonight, you'll know what DNA is made of, how it stores information, how it's read, how it's passed on, and how it connects you to everything that's ever been alive. You'll see why it matters, and why, ultimately, it doesn't need to. DNA would be doing its job just fine whether you understood it or not. This leads us to consider your own experience, and how this invisible molecule has shaped everything about you without you ever noticing. You've probably heard people say things like, "It's in your DNA," usually after someone shows an alarming fondness for spreadsheets or an inability to reverse a car. It's become a casual phrase, but behind it is something very real. DNA is quite literally inside you, in every cell, influencing almost everything about you, whether you've asked it to or not. Look in the mirror. The shape of your nose, the curl of your hair, the color of your eyes. All of that came from instructions written long before you were born. You didn't choose them. You just inherited them. And you'll pass on some version of those instructions whether you mean to or not. It's not destiny, exactly, more like stubborn paperwork that keeps filing itself. You might have noticed family resemblances. Maybe you've got your dad's ears or your grandmother's chin, or perhaps your second toe is longer than your big toe. And, unfortunately, so is your uncle's. These patterns aren't just coincidences. They're signs that something is being copied, passed down, repeated across generations like a genetic group chat that no one can leave. It's not just about looks, either. DNA influences things like how your liver processes chemicals, how your brain builds memories, and how your muscles respond to exercise. It plays a role in whether coriander tastes fresh or like soap. Some people blame DNA for not liking broccoli. Others blame it for liking reality TV. Both are probably coping mechanisms. But here's the strange thing. All of these deeply personal traits come from something you can't see. You've never looked at your DNA. You've never felt it working. And yet, it's there, quietly active, copying itself inside you right now, every second, without pause. It never takes a tea break, which is frankly rude. We think of ourselves as individuals, and we are. But at the molecular level, your body is running a program written long ago, updated slightly over time, and carried forward from people you've never met. Your great-great-grandparents had a hand in your freckles, which is either touching or mildly annoying, depending on your freckles. So before we talk about what DNA is in detail, it helps to realize just how much of your everyday experience comes from it. The way you grow, the way your organs work, the way your cells replace themselves after you've scraped your knee. All of it shaped by this quiet molecular script humming away in the background. This leads us to consider what we actually observe about ourselves. and how those first impressions, though familiar, might not tell us the full story. Every day, you see evidence of inheritance all around you, without needing a microscope or a biology degree. A child has their mother's smile. Siblings share similar dimples. Twins, identical or not, seem like variations on a theme. These patterns appear obvious, familiar. They suggest something is being passed on from parent to child, something quietly consistent. You might describe it as being in the blood, or simply say, she takes after her father. But what we actually see is just the surface: hair texture, eye color, height, all observable traits. It's tempting to believe these appearances are the explanation, that what you see tells you everything you need to know about where it came from. It doesn't. Underneath those visible patterns is a level of detail far beyond human sight. You can't watch DNA at work with your eyes. You can't feel it pulsing through your cells. You might not even notice it when it goes slightly wrong. Until you start developing hay fever every spring and wondering which side of the family to blame. Nature isn't exactly subtle, but it is quiet. For centuries, people used guesses and folk logic to explain inheritance. Maybe the father's strong features cancelled out the mother's. Maybe traits skipped generations because they fancied a holiday. Without tools to see what was really going on, explanations lean towards the poetic, the mystical, or just the confidently wrong. Human beings are quite good at that. Even today, much of what we think we know about genetics comes from simplified stories. You might think of DNA as a kind of barcode, or a list of boxes that get ticked. In truth, it's more like a very long instruction manual, where most of the pages are still being argued over by the scientific equivalent of a particularly sleep-deprived book club. What we observe in life is a hint, sometimes accurate, sometimes misleading, about what's happening deep inside our cells. Identical twins might look the same but behave differently. Siblings can share DNA, but have completely different reactions to the same illness. The surface, it turns out, is not the whole story. It's barely the first sentence. To truly understand DNA, we need to go beyond what our eyes can show us, past the level of the skin, the organs, even the cells. We need to go deeper into the realm where molecules carry information, copy themselves, and somehow produce the blueprint for a working human being, or at least one capable of finishing their GCSEs. This leads us to consider the scale at which DNA exists, and just how far it lies beyond our natural sense of sight or touch. To understand DNA properly, we need to shrink things down, quite a lot. Your body contains trillions of cells, and inside almost every one is a copy of your DNA. But to actually see it, even with the world's best microscope, you'd need to zoom in beyond anything your eyes were designed to handle. Human senses simply aren't built for this scale. A single cell is already too small for us to see without help. Inside that, you'll find a nucleus. Inside that, you'll find long molecules of DNA. Stretched out, they'd reach about 2 meters in length, yet somehow crammed into something far smaller than a grain of sand. It's like fitting a paperback novel inside a postage stamp. On both sides, the structure of DNA exists at the nanometer scale. That's 1/1000th of a meter. For reference, a single human hair is about 80,000 to 100,000 nanometers wide. DNA? Just 2 nanometers across. It's the sort of scale where atoms become visible, not as round balls, but as arrangements of charge and probability. Yes, at this level, even certainty starts to wobble. And yet, this microscopic scale contains instructions that shape giraffes, jellyfish, and GCSE students who forget their homework. All made possible by a pattern of molecules organized into a code. The sheer contrast between size and influence is difficult to grasp. Something so small carrying the information to make, well, everything that eats, breathes, or complains about Mondays. To help visualize it, imagine a library where every book contains instructions for making a living thing. Then shrink each book to the size of a dust mote. That's roughly the scale we're dealing with. Except DNA isn't made of paper, it's made of repeating chemical units. Small, precise molecules joined together in long chains. Chains which, somehow, encode life. Despite how abstract this might sound, it's not magic. DNA is just chemistry, but chemistry done with almost ludicrous precision. Bonds form, shapes twist, patterns emerge, all guided by basic physical forces. These interactions don't need supervision or opinion. They happen automatically, which is more than can be said for group projects. This is also where our usual intuition breaks down. We're used to thinking in centimeters, meters, hours, maybe years. But DNA operates far below those everyday measures. The molecules in your cells are forming and unforming constantly, with a speed and scale that feels completely detached from normal life. Because, quite simply, it is. This leads us to consider how these tiny molecules actually work, and what kind of instructions they carry inside their microscopic spiral. Now that we've zoomed in to the right scale, let's look at what DNA actually is. Not in abstract terms, but in chemical reality. It's a molecule, a very long one, made from smaller repeating units called nucleotides. Each nucleotide contains 3 parts: a sugar, a phosphate group, and one of 4 chemical bases: adenine A, thymine T, cytosine C, or guanine G. Not exactly catchy, but they get the job done. The DNA molecule forms a shape called a double helix. It looks a bit like a twisted ladder. The sugar and phosphate make up the sides, and the bases pair up to form the rungs. Each base has a specific partner. A always pairs with T, and C always pairs with G. No exceptions, no debates, just strict molecular preferences. A sort of chemical matchmaking service with absolutely no room for romance. What's important here is the order of those bases. The sequence A, G, C, T, and so on carries information. Change the order, and you change the meaning. In DNA, that order forms instructions for building proteins, which do most of the work in your body. Muscles, enzymes, hormones, all protein-based, all built according to those coded instructions. You could imagine it like a recipe book, where each gene is a specific recipe. Some are for ingredients like keratin used in hair and nails, others for collagen found in skin and joints. A few are just sitting there doing nothing, like that cookbook section you keep meaning to try, but never do. Scientists still argue over whether those unused bits are truly pointless, or just misunderstood. These instructions are stored in long strands called chromosomes. Humans have 23 pairs, each inherited from one parent. Your chromosomes are tucked inside the nucleus of almost every cell in your body. And somehow, that tiny space holds about 3 billion base pairs. It's like storing all of Wikipedia in something the size of a salt crystal, though arguably, DNA's accuracy rate is a bit better. Despite this complexity, DNA doesn't act with intention. It doesn't try to do anything. It's chemistry following physical laws. Bases bond in predictable ways, sequences are copied according to rules, and proteins are built because the system just works. No magic, no agenda, just molecules doing what molecules do. On autopilot. The elegance lies in the reliability. With just 4 chemical bases, life builds everything, from plankton to parrots. It's hard not to appreciate the efficiency. DNA isn't flashy, but it's dependable. Like the sort of person who brings a packed lunch to every school trip. Not glamorous, but everyone benefits. This leads us to consider how this process unfolds in detail, how DNA gets read, copied, and turned into the actual parts that make life happen. We've said DNA holds instructions, but instructions only matter if something can read them. Inside your cells, that something is a complex and staggeringly efficient molecular system. It starts with transcription. A stretch of DNA, let's say a gene, is copied into a temporary molecule called messenger RNA. This happens inside the nucleus, using enzymes that match up base pairs with ruthless consistency. The messenger RNA, or mRNA for short, is like a photocopy of a page from the instruction manual. It doesn't copy the whole genome, just the bit that's needed. That copy then exits the nucleus and heads for a ribosome, which is essentially a protein-building machine. It reads the RNA code 3 letters at a time. Each triplet, called a codon, tells it which amino acid to add next. Amino acids are the building blocks of proteins. There are 20 different types, and the order in which they're linked determines the shape and function of the resulting protein. The ribosome strings them together, folds them into complex shapes, and voila! You've got a protein ready to do a specific job. Maybe it's carrying oxygen, or speeding up a chemical reaction, or just making your skin stretchy. The whole process, DNA to RNA to protein, is often summarized as the central dogma of molecular biology. It's not a particularly catchy name, but it's stuck. This isn't a one-time event either. It's happening all the time. Right now, millions of your cells are carrying out transcription and translation while you sit there, possibly forgetting where you left your charger again. Let's simplify it further. DNA is like a master cookbook. RNA is a handwritten copy of one recipe. The end product is a protein, ready to be used by the body. Except this cook works 24/7, never complains, and doesn't mess about with garnish. This system has to be accurate, but not too rigid. Mistakes can lead to problems, but small changes over time, mutations, are also how evolution happens. So it's a delicate balance between fidelity and flexibility. And it all unfolds on a scale so small, you'd need high-powered lab equipment just to watch it. Which feels unnecessarily secretive for something that's literally building your spleen. And still, it's not directed. No one tells the cell which genes to activate. Chemical signals, environmental cues, and internal feedback loops trigger the whole process. It's more like a chain reaction than a choreographed performance. Things happen because the conditions allow them to, not because someone told them to. This leads us to consider how this molecular machinery isn't just a human feature, but something found across nearly all life, from bacteria to blue whales. The process we've just described, DNA being read, copied, and turned into proteins, isn't unique to humans. It's not even specific to animals. The same mechanism occurs in plants, fungi, bacteria, and just about everything else we currently consider alive. DNA doesn't have a loyalty program. It simply works. So nature uses it everywhere. Whether you're looking at an oak tree, a blue whale, or a colony of E. coli living on your kitchen sponge, the basic method is the same. DNA carries instructions. RNA delivers them. Proteins do the work. It's astonishingly consistent. A banana shares around 60% of its genes with you, which, depending on how your day's going, might not come as a shock. This shared system points to something deeper, a common origin. The fact that all living organisms use the same 4 bases, the same codon rules, and the same basic flow of information suggests life on Earth didn't invent DNA multiple times. It invented it once, and that method was so effective, nothing better ever came along. Efficient, unglamorous, and irritatingly robust. This doesn't mean every organism is the same. Far from it. Genes are switched on or off in different ways, proteins are modified, and entire systems of regulation exist. But the foundation is consistent. And it doesn't matter whether you're a human applying moisturizer or a bacterium drifting in deep-sea vents, your cells follow the same molecular instructions. This consistency is useful. It means scientists can study DNA in fruit flies or mice and still learn things about human biology. It means you can insert human genes into bacteria to produce insulin. Yes, we do that. We give E. coli human DNA, and it politely makes medicine. No fuss, just biology doing its thing. But more than usefulness, this sameness points to something important about nature. The laws of chemistry and physics don't change depending on the species. They don't care whether you're conscious, cute, or capable of using cutlery. DNA works because the underlying rules are universal and completely indifferent. It's a humbling thought. The instructions that built you are made of the same chemical logic as those in a blade of grass. Not worse, not better. Just different combinations of the same building blocks, arranged by natural processes over time. If you were expecting something more exclusive, apologies. Nature's quite fond of bulk ordering. This leads us to consider how these molecular processes play out, not just across species, but across vast stretches of time. Through the long, patient timeline of life on Earth. DNA isn't just a code used across species. It's a record of time. Every strand in every cell carries traces of the past. Not in dates or diaries, but in mutations, patterns, and preserved sequences. DNA changes, slowly and unevenly, over generations. And through those changes, we can glimpse a history that stretches back billions of years, long before humans were involved. A mutation is a change in the DNA sequence. It might swap a letter, delete one, or insert a new bit. Most of the time, these changes do nothing noticeable. Occasionally, they cause disease. Very occasionally, they improve survival. But whether useful or not, they accumulate, they're passed on. And they add up to something far more powerful than any individual life. Using these changes, scientists can trace ancestry, not just between individuals, but between entire species. Humans and chimpanzees share around 98.8% of their DNA. With mice, it's about 85%. These numbers aren't based on guesswork. They come from comparing genomes and measuring how much time has passed since two species last shared a common ancestor. And then there's the deeper timeline. DNA evidence shows that all life shares a common origin. A single, early form of life, likely emerging over 3.5 billion years ago, gave rise to everything alive today. That's longer than continents have held their shape. DNA has been copied, changed, and refined across eons, drifting steadily forward through deep time. We tend to think in short bursts, school years, work weeks, waiting for Wi-Fi to reconnect. But DNA works across unimaginable stretches. Whole species rise and fall, ecosystems shift, planets cool. Yet the same basic code continues, moving from one form of life to the next, rewriting itself with no final destination in mind. Just ongoing replication and survival. Even within your own body, DNA's sense of time is strange. You carry mutations your parents didn't have. You pass some on without noticing. Tiny genetic changes inch their way through generations, shaping future outcomes without plan or awareness. The process is slow, automatic, and astonishingly consistent for something that seems to involve so little effort. Looking at DNA is a way of looking backwards. It holds echoes of ancient viruses, fragments of genes long switched off, and shared sequences with long extinct relatives. It doesn't explain everything, but it reveals a quiet, cumulative truth. Life moves through time by copying itself, imperfectly, persistently, and without needing to understand what it's doing. This leads us to consider how all of this, the chemical code, the copying, the time, exists without concern for us, and what it means to live in a universe where the rules apply with beautiful, total indifference. DNA doesn't know who you are. It doesn't care what you want. It doesn't have goals, dreams, or a favorite biscuit. It's not here to make you happy, successful, or emotionally fulfilled. It simply copies itself. Over and over. Sometimes perfectly, sometimes with small mistakes. It does this not because it should, but because chemistry makes it possible. That might sound bleak, but it's one of the reasons DNA is so reliable. It isn't guided by opinion. It doesn't respond to trends or feelings. There's no hidden meaning behind which traits get passed on. A gene that helps survival tends to stick around. One that doesn't. There's no cosmic complaint box, just chemical consequences played out across time. You're here not because DNA intended it, but because previous copies happened to work well enough to keep going. The process doesn't reward cleverness or kindness. It rewards persistence. If something works, by accident or otherwise, it continues. If it doesn't, it stops. Nature isn't being cruel, it's just being entirely disinterested. This indifference is what makes evolution possible. Mutations arise randomly. Some help, some harm, most do nothing. The environment selects which versions are more likely to survive. But the environment, too, has no agenda. It changes, it shifts, and living things either keep up or they don't. DNA responds, not by choice, but by consequence. That's the strange truth. You exist because of countless past accidents that happened to survive. Your DNA is shaped by floods, famines, ice ages, and microscopic changes that played out without warning or foresight. You are, in effect, the temporary arrangement of atoms that happened not to get deleted. It's not exactly flattering, but it is accurate. And despite all that, DNA produces things that think. It builds eyes that see, brains that wonder, and fingers that point at the stars. Not because it meant to, but because those traits happen to help. Life's most remarkable qualities weren't planned. They emerged from systems that didn't know or care what they were doing, which, frankly, sounds like most group chats. The comfort here isn't in being important. It's in knowing that importance isn't required. DNA doesn't wait for your approval. It does what it does, regardless. And somehow, that process gave rise to every creature, every tree, every cell that's ever lived. not because the universe wanted it, but because the rules allowed it. This leads us to consider how humans came to understand any of this. How, over centuries, we slowly revealed what DNA is, how it works, and what it tells us about life itself. Long before DNA was discovered, humans had already noticed something strange about how traits were passed on. Parents and children often looked alike. Certain diseases seemed to run in families. Farmers and breeders saw patterns in animals and crops. Tall plants made more tall plants. Curly-tailed pigs stayed curly-tailed. Clearly, something was being inherited, though no one had any idea how. Ancient cultures came up with all sorts of explanations. Some believed traits came from the father's blood. Others thought the mother's womb shaped the child like clay. There were ideas about vital forces, essences, and humors. Not terribly useful, but to be fair, they were working without microscopes or molecular biology, just pattern recognition and guesswork. The Greeks had a go at it, of course. Hippocrates suggested tiny particles carried information from all parts of the body to the reproductive organs. Aristotle disagreed and said the father's contribution was like a sculptor shaping the passive material provided by the mother. Not exactly accurate, but again, he didn't have a PCR machine, just a beard and some opinions. What these early thinkers lacked in tools, they made up for in patience. Over time, careful observers began recording details. How often traits appeared, which combinations worked, and which ones didn't. In agriculture, this became practical knowledge. In humans, it became the awkward explanation for why Uncle Peter still looks like great-granddad, despite no one asking him to. For centuries, the key idea was that inheritance existed, but its mechanism was a mystery. Something was clearly being passed on, but whether it was fluids, vibrations, or a mystical soul packet was very much up for debate. It's worth remembering, most people throughout history had no reason to question this. They saw the outcomes, and that was enough. There were also many false starts. Ideas like blending inheritance suggested that a child was a simple average of its parents. Sounds logical. Until you realize that, if that were true, extreme traits would vanish over generations. Instead, they persist. A reminder that things that sound obvious often aren't. Much like the idea that trousers with more pockets are automatically more useful. Still, the accumulation of observations mattered. Slowly, very slowly, the patterns began to outgrow the explanations. Farmers didn't need to understand nucleotides to breed better wheat. Midwives didn't need chromosomes to spot family resemblances. But the groundwork was being laid, an unintentional prelude to science. This leads us to consider the moment when those vague observations began to harden into scientific insights, and when someone finally asked not just what was passed on, but how. The real shift began in the mid-1800s, not in a grand laboratory, but in a quiet garden. Gregor Mendel, an Augustinian monk with a fondness for pea plants and a tolerance for repetition, began crossing different varieties and keeping meticulous records of the results. It wasn't glamorous, but it was precise, and that patience changed everything. Mendel observed that traits like flower color and seed shape didn't blend. They appeared in distinct patterns, generation after generation. He counted how often they showed up, and realized something predictable was happening. Traits were inherited as separate units, later called genes. He didn't know what genes were made of, but he could see how they behaved. From these observations, Mendel developed the idea of dominant and recessive traits. A dominant trait would show up if present. A recessive one could hide, only appearing if both copies were the same. It was simple, mathematical, and repeatable. And crucially, it didn't match the blending theories most people believed at the time. The brilliance of Mendel's work wasn't just the idea, it was how he showed it with data. He ran experiments with thousands of plants. He tracked generations. He wrote it all down. It was biology by numbers, long before genetics even had a name. And naturally, it was completely ignored for decades. Because of course it was. At the time, no one connected his work to anything larger. The word gene hadn't even been invented. DNA hadn't been discovered. And Mendel, being a monk with a quiet job, wasn't exactly storming academic conferences. His paper drifted into obscurity, appreciated by approximately 4 people, possibly including his mum. But Mendel laid the foundation. Without microscopes, without molecular models, he'd shown that heredity followed patterns. That something inside living things, something invisible, was controlling traits with consistency. It was the first real clue that the laws of inheritance weren't mysterious forces, but rules that could be studied, tested, and explained. It would take decades before anyone realized how right he'd been. When scientists finally rediscovered Mendel's work in the early 1900s, they connected it to the emerging study of chromosomes and cell division. Suddenly, heredity had a structure. Not just theory, but a place in the cell. And the race to uncover its physical basis had quietly begun. This leads us to consider how scientific tools, more powerful eyes and sharper methods, helped reveal what those units of inheritance actually were and where they lived inside the cell. Once Mendel's work was dusted off and taken seriously, scientists began hunting for the physical location of these units of inheritance. But seeing something as small and delicate as DNA required more than curiosity. It required tools, new instruments that could stretch human senses far beyond their natural limits. Biology had to start building better eyes. One of the key breakthroughs came with the development of better microscopes. Using dyes and staining techniques, researchers were able to see structures inside cells more clearly. They noticed thread-like forms during cell division. These were chromosomes. And soon, they realized that these structures behaved exactly as Mendel's traits did, dividing, pairing, separating, suspiciously well behaved, for something so tiny. Scientists began associating genes with specific positions on these chromosomes. The chromosome theory of inheritance was born. But even then, no one knew what chromosomes were actually made of. There were proteins in there, and a mysterious substance called nuclein, later identified as DNA. Most people assumed the proteins were doing the hard work. DNA, being made of just 4 repeating parts, seemed far too boring to matter. The next step came from chemistry. In the early 20th century, biochemists began isolating DNA and studying its chemical structure. They identified the 4 bases, A, T, C, and G, and noticed something peculiar. In every DNA sample, the amount of A always matched T, and C always matched G. This hinted at a pattern, but no one yet understood the full structure. Then came X-ray crystallography, a method for studying molecules by firing X-rays at them and analyzing the resulting scatter. It was tedious, precise, and required great patience. Rosalind Franklin, one of its most skilled practitioners, produced images that hinted at a helical shape, a twisted ladder. It was a breakthrough hidden in shadows and angles. Centrifuges were also key. These machines could spin biological samples at high speeds, separating components by weight. DNA, it turned out, was heavier than proteins. And when you broke cells apart and spun them down, you could isolate it. White, stringy, and not particularly glamorous. Like pulling invisible spaghetti out of soup, if the soup were full of nuclei. All of these tools work together. Stains to see cells, chemistry to analyze molecules, X-rays to reveal structure, centrifuges to separate parts. No single technique gave a complete answer. It was the slow, layered combination that gradually revealed what DNA was, where it lived, and how it might hold information. Science by accumulation and stubbornness. This leads us to consider what happens when those early assumptions go wrong, and how scientific progress often means realizing we've misunderstood something completely. Science rarely moves in a straight line. For every accurate conclusion, there are usually a few confident missteps along the way. DNA, now known as the carrier of genetic information, was once dismissed as too boring to matter. For years, scientists assumed proteins, complex, variable, and mysterious, were the real stars of inheritance. DNA was thought to be little more than molecular scaffolding. This assumption made sense at the time. Proteins are built from 20 different amino acids, while DNA is made from just 4 bases. It seemed unlikely that something so repetitive could hold detailed instructions, like expecting someone to write Shakespeare using only 4 letters. But nature, as it turns out, is both more efficient and less interested in human expectations. The shift began with a series of quiet but decisive experiments. In the 1940s, Oswald Avery and his team showed that DNA, not protein, was the transforming principle, able to transfer traits between bacteria. Many biologists didn't believe it. They weren't ready to let go of their protein obsession. You could say the data was ahead of its time, or just that people don't like admitting they're wrong. Then came the Hershey-Chase experiment in 1952. Using viruses that infect bacteria, they labeled DNA with one type of radioactive tag and proteins with another. Only the DNA entered the bacteria. The conclusion was unavoidable. DNA, not protein, was passing on the instructions. At this point, it became very difficult to keep pretending otherwise. Science depends on correction. That doesn't mean it's always quick. People held on to flawed ideas because they were familiar or fit the assumptions of the day. This isn't unusual. The history of science is full of ideas that were cherished right up until they were proven false, like geocentrism, or phrenology, or low-fat crisps. The DNA story also reminds us that even correct ideas can be misleading if oversimplified. For example, the one gene, one protein model seemed neat but turned out to be wrong. Some genes make multiple proteins. Some DNA doesn't code for proteins at all. Some bits regulate other bits. It's not chaos, but it's far messier than the textbooks used to suggest. Even today, many people think of DNA as a perfect blueprint. In reality, it's more like a very detailed instruction manual, with handwritten notes in the margins, pages that contradict each other, and the occasional sentence that no one understands. Accurate enough to build a human, but not so tidy that it couldn't benefit from an editor. This leads us to consider how scientists actually tested these ideas, how experiments, carefully designed and repeated, helped confirm the role of DNA and uncover the details of how it behaves. After realizing DNA was the molecule of inheritance, the challenge became proving how it worked, not through speculation, but through repeatable, testable experiments. This wasn't guesswork or hopeful theorizing. It was slow, careful observation of molecules doing their job, one base pair at a time. Welcome to the lab, where progress smells faintly of ethanol and regret. One of the first goals was to understand how DNA copies itself. Scientists hypothesized a semi-conservative model, where each new DNA molecule would keep one strand from the original and make a new complementary one. The famous Mendelssohn-Stoll experiment in 1958 confirmed this. They used nitrogen isotopes, one heavy, one light, to trace the origin of each DNA strand. The result? A neat pattern showing DNA's ability to copy with a built-in memory. To test how DNA turns into protein, scientists had to crack the genetic code. They created synthetic RNA with repeated bases, UUU, AAA, etc., and injected them into cell extracts. The resulting proteins showed which codons matched which amino acids. This work, led by Nirenberg and others, revealed the codon table, a clear, three-letter mapping between DNA and the proteins it builds. Like finally finding the key to a very specific and slightly passive-aggressive substitution cipher. Of course, many experiments weren't that tidy. Discovering how genes were turned on or off, regulated, took decades. The lac operon in bacteria became a model system. Scientists found that certain proteins could bind to DNA and block or trigger gene expression, depending on the conditions. The result wasn't just DNA makes protein, but DNA makes protein when necessary, which is far more realistic. Much like humans only making tea when a certain threshold of stress is crossed. Precision was everything. Experiments had to isolate one variable at a time, change just one base, knock out one gene, watch one protein. It's not glamorous work, often repetitive and full of failure. But each step reveals a sliver of understanding. And when hundreds of these experiments are pieced together, they form the scaffolding for everything we now know about molecular biology. What made these tests so powerful was that they could be repeated. Different labs, different countries, same outcome. That's the strength of science, not flashy results, but reliable ones. The sort of consistency that builds planes, diagnoses illnesses, and occasionally proves your biology teacher was right all along. Unfortunately, through these experiments, DNA shifted from a theory to a working model, something scientists could predict, manipulate, and rely on. It wasn't just a strange molecule anymore. It was an operating system for life. And, like most operating systems, understanding it involved quite a few forced restarts. This leads us to consider how these discoveries didn't come from isolated geniuses, but from collaboration. Thousands of researchers contributing over time, refining each other's work and building a shared understanding of how life really functions. Science likes to celebrate individuals, those headline names that get attached to breakthroughs. But the truth is, most discoveries about DNA didn't come from lone geniuses in dramatic moments of insight. They came from groups of researchers working across borders, decades, and disciplines, a kind of long, quiet relay race, only with more pipettes and fewer medals. The structure of DNA is often credited to Watson and Crick. They did publish the famous double helix model in 1953. But their work relied on data from Rosalind Franklin's X-ray crystallography, Maurice Wilkins's collaborative support, and a good deal of information sharing that was, let's say, not entirely straightforward. The Nobel Prize didn't go to Franklin. She had died before it was awarded. And science, like the Oscars, does have a habit of missing people in the credits. Beyond the double helix, discovery continued. Across the 20th century, labs in the United States, the UK, Germany, Japan, and many others contributed piece by piece, uncovering replication mechanisms, refining the genetic code, identifying the enzymes that splice, repair, and copy DNA. Each group added their own fragment of knowledge to the puzzle. There was no single eureka moment, just a steady flood of careful, detailed progress. Much of this work was shared through journals, conferences, and a surprising number of international collaborations. Scientists from vastly different cultures and backgrounds came together over a common language: data. A base pair is a base pair, whether you're working in Cambridge or Kyoto. Though your lab snacks may differ, this collaborative approach wasn't just polite, it was necessary. Biology is too complex, and the tools too specialized, for any one person to tackle it alone. One lab might specialize in sequencing techniques, another in molecular modeling, another in bacteria that refused to behave as predicted. Sharing findings meant faster progress and fewer repeated mistakes. In theory, anyway. Even the Human Genome Project, launched in the 1990s, was built on international teamwork. The goal was ambitious: map all 3 billion base pairs of human DNA. No single country could do it alone. Labs from across the world divided the work, standardized their methods, and compared notes. It wasn't fast, and it wasn't always friendly, but it worked. By 2003, the first full sequence was complete. All of this shows that understanding DNA isn't about one discovery. It's about many minds chipping away at the same question. Knowledge doesn't belong to individuals. It accumulates, adapts, and spreads. Much like DNA itself, really, though with fewer transcription errors. Most of the time, this leads us to consider the precision we've now achieved, how far our tools have come, how clearly we can now read and edit DNA, and what that means for the questions we're still trying to answer. Today, our ability to study DNA is nearly unrecognizable from the early days of microscopes and handwritten notebooks. What once took years can now take hours. Entire genomes, billions of base pairs, can be sequenced by machines smaller than your suitcase. It's not quite magic. It's better. It's reliable technology built on decades of scientific persistence and, presumably, a lot of grant applications. Modern sequencing methods, like next-generation sequencing (NGS), allow scientists to read huge amounts of DNA at once. Instead of decoding one gene at a time, we can now sequence everything in parallel, like copying every book in a library at the same time. It's fast, detailed, and far more accurate than the early, painstaking, letter-by-letter approaches. And unlike most group projects, it actually works under pressure. This precision isn't just about reading. We can now write changes too. CRISPR-Cas9, a gene editing tool first described in the early 2010s, lets scientists cut DNA at specific points and insert, delete, or tweak particular sequences. It's based on a natural defense system in bacteria, which, unsurprisingly, turned out to be better at genetic editing than most humans with scissors. With CRISPR, researchers can switch off faulty genes, correct inherited disorders in lab models, and explore how specific mutations affect traits. The scale of control is extraordinary. It's not theoretical, It's being used right now in clinical trials and research labs around the world. And occasionally, someone suggests using it to create glow-in-the-dark pets. Because of course they do. Beyond human genetics, we're sequencing and editing plants, bacteria, viruses, and endangered species. Genomes from ancient fossils are being reconstructed. Microbial DNA is being found in deep-sea vents and Arctic permafrost. The technology doesn't just show us what's here now, it lets us reconstruct the past and imagine futures, some more sensible than others. Yet despite all this control, the more we see, the more complicated it becomes. Genes interact in tangled networks. One gene can affect many traits. One trait can be influenced by hundreds of genes. DNA is just the start. Epigenetics, environmental triggers, and timing all shape what actually happens in a living body. Reading the letters is easy now. Understanding the grammar, still a work in progress. What's impressive is how quiet all this power is. These techniques operate on invisible molecules inside living cells, changing their behavior without fanfare. No sparks, no flashing lights. Just an edited base, a nudged protein, a corrected line of code. It's the biological equivalent of proofreading reality, which frankly is long overdue. This leads us to consider what we still don't know. Because even with all this precision, DNA remains full of puzzles, mysteries that resist tidy answers, and force us to admit that we're only just beginning to understand how life truly works. Even with all the technology, cleverness, and highly caffeinated researchers, DNA still hasn't handed over all its secrets. Yes, we've sequenced genomes, edited genes, and mapped regulatory pathways. But there are still vast stretches of uncertainty, regions of DNA whose functions we don't fully understand. In other words, we've read the book, but some pages seem to be in a language no one speaks. Take non-coding DNA. Only about 1–2% of our genome actually codes for proteins. The rest used to be dismissed as junk DNA. Turns out, it's not junk. Much of it plays subtle roles in regulation, structure, and timing, but quite a bit of it still defies explanation. It's like finding a massive appendix in a manual and realizing some of it might be instructions, and some might just be there to confuse you. Then there's epigenetics, chemical tags that sit on top of the DNA and influence how genes are expressed without changing the sequence itself. These tags can respond to the environment and in some cases even be passed down to offspring. This suggests that your genes aren't your fate, they're more like guidelines, occasionally rewritten by stress, diet, or whether your nan smoked during the war. We also don't fully understand how genes interact with one another. A single gene might contribute to dozens of traits, depending on when and where it's active. And the same trait, like height or the ability to digest milk as an adult, might involve hundreds of tiny genetic factors working together. It's less like a straightforward recipe and more like 17 chefs whispering contradictory instructions at once. Another puzzle is the origin of DNA itself. We can study how it behaves now, but how did such a complex, self-replicating molecule emerge from the chaos of early Earth? There are hypotheses involving simpler molecules like RNA, which may have come first. But we're still piecing together the steps from chemistry to biology, and the pieces are not exactly snapping into place. And then there are the quirks. Why do some plants have genomes 10 times the size of ours? Why do certain bacteria keep spare genes they never use? Why do identical twins sometimes develop different illnesses. These aren't just oddities. They're reminders that life is more flexible and more confusing than our categories suggest. It's also unclear how far we should go with what we do understand. The ethics of gene editing, data privacy in genome sequencing, and the limits of biological control are all still being debated. Knowing something doesn't mean we're ready to use it wisely, especially when it comes to editing living organisms, where the margin for error is small and the consequences not always predictable. This leads us to consider DNA on a much larger stage, not just in labs and clinics, but out across time and space. Because to fully understand where DNA fits in the universe, we need to look at its role in the broader story of life on Earth, and how it might help us find life beyond it. If DNA is the basis for life on Earth, it raises a simple question. Does something similar exist elsewhere? We've spent decades scanning the skies, probing planets, and analyzing meteorites, hoping to spot anything that even vaguely resembles life. So far, silence. Or at least, a lack of strongly worded invitations from nearby galaxies. But that doesn't mean life isn't out there. It just means we haven't found the right clues yet. And if alien life does exist, we don't actually know whether it would use DNA at all. DNA might be one solution to life's challenges, not the only one. There could be other molecules that store and pass on information. Different shapes, different chemistry, same job description. Still, there are good reasons to expect similarities. DNA works well. It's stable, compact, and efficient. It stores huge amounts of information in a small space and can be copied reliably. In fact, some researchers think molecules with similar properties, long chains with repeating units, capable of folding and replicating, might be common in any universe with carbon, water, and a bit of patience. So, where might we look? Mars is still a favorite candidate. It's dry and dusty now, but once had water. We found organic molecules in its soil. Though organic in chemistry just means contains carbon, not alive, and enjoying brunch. Europa and Enceladus, moons of Jupiter and Saturn, have icy crusts and liquid oceans beneath. If you're a microbe, that might be a perfectly acceptable postcode. There's also Titan, with its methane lakes and thick atmosphere. It's cold enough to freeze your eyebrows off, but some think alien biochemistry could still operate there. Just not the kind we're used to. And of course, there are exoplanets. Thousands of them now discovered orbiting distant stars, some in the so-called habitable zone. Though habitable here usually just means not instantly deadly. To find life or rule it out, we'll need to look for biosignatures, chemical traces that hint at biology. Oxygen, methane, or complex organics in the right ratios might suggest something is alive and breathing. But these signals can be caused by geology too, so it's a bit like trying to guess who lives in a house based on the smell of the bin. Meanwhile, Earth gives us some perspective. Life here is astonishingly adaptable. Bacteria thrive in boiling acid, deep beneath the sea, and inside nuclear reactors, which suggests that if life can emerge, it probably will. It just might take forms that surprise us, or make us feel rather ordinary. This leads us to consider the future of DNA. How far we might go in rewriting it, reinventing it, or even designing entirely new forms of life from scratch. If nature took 4 billion years to get here, we're now at the point where we might take over the pen. Tonight, we find ourselves not just observing DNA, but rewriting it. For most of history, We've been at the mercy of evolution, waiting for mutations, hoping something useful might happen. Now, thanks to modern tools, we can cut, copy, and paste DNA with almost irritating precision. It's less survival of the fittest and more editing by committee. CRISPR is the star of this show. It's a gene editing tool that works like molecular scissors. If your scissors came with a GPS and a grudge, using it, scientists can target specific sections of DNA and either snip them out or replace them entirely. It's efficient, cheap, and, depending on who you ask, a little bit terrifying. We've already used CRISPR to treat diseases in humans, modify crops, and engineer animals. Some researchers have used it to make mushrooms that don't brown, or pigs with organs more suitable for transplant. It's practical biohacking. We're not creating superheroes yet, but we are learning how to fine-tune life, one base pair at a time. Beyond editing what exists, we're also building from scratch. Synthetic biology aims to design new life forms with entirely artificial DNA. Some labs have already created cells with extra letters in their genetic alphabet. Not just A, T, C, and G, but new ones that nature never invented. Like adding extra symbols to your keyboard, just in case life wasn't complicated enough already. This raises a rather large question. If we can design DNA, are we still discovering life, or are we now inventing it? The boundary between biology and engineering is getting fuzzier by the year. Organisms are becoming platforms. Cells are being treated like hardware. And somewhere, a yeast cell is probably being forced to manufacture vanilla flavoring for your ice cream. Of course, with great power comes, well, regulation. Gene editing in humans is still controversial. The idea of designer babies makes most people uncomfortable, and for good reason. There are ethical, social, and biological consequences we don't fully understand. Just because you can give your child perfect pitch doesn't mean you should, especially if you then have to hear them sing. But despite the hype and the fears, most of this technology is still in its early stages. Editing genes in a living organism is difficult. Predicting long-term effects is even harder. Nature has had billions of years to test its code. We've had about 15 minutes and a very excitable press release. This leads us to consider a bigger question still, not just how we alter DNA, but what that tells us about being alive in the first place. Where does life end and machinery begin? The more we tinker with DNA, the more it raises a strangely personal question. What exactly counts as life? If we replace enough parts of a cell with artificial ones, is it still alive, or just very committed cosplay? At some point, biology and engineering stop being separate fields and start becoming a very confusing Venn diagram. Let's start with synthetic cells. Scientists have already built simple cells in the lab with minimal genomes. Stripped-down organisms that do only what's necessary to survive and reproduce. It's like designing a mobile phone that only texts and makes calls. Unfashionable, yes, but efficient. These cells can't do much, but they do meet the basic definition of life. Then there's xenobiology, research into life forms with altered biochemistry. Some teams are developing organisms that use synthetic DNA, or even entirely different molecules instead of DNA and proteins. They still grow, evolve, and reproduce, but they do it with ingredients not found in nature. It's life, but not as we know it. Spock would be thrilled. Add to this the growing use of biocomputers, living cells that store data or run logic circuits. One bacterium has been engineered to act like a camera, storing a black and white image in its DNA. Slightly creepy, but undeniably clever. Cells are no longer just living things, they're also tools, sensors, and platforms for programming. Meanwhile, some researchers are working on non-biological systems that still tick many of the boxes for life. Chemical droplets that grow and divide. Molecular machines that respond to their environment. They don't have DNA, but they show lifelike behavior. The line between living and non-living is looking less like a solid wall and more like an unconvincing garden fence. This matters because it forces us to re-examine our definitions. Life used to mean carbon-based organisms with cells, DNA, metabolism, the usual checklist. But now, with artificial cells, digital biology, and lab-grown hybrids, we're having to ask, is life a set of ingredients, or is it a process? A function? A pattern? There's also the philosophical question of identity. If we replace enough of our own biology with engineered parts, synthetic organs, neural implants, maybe even programmable genomes, are we still human? Still alive? Or just increasingly elaborate hardware with a fondness for toast? For now, most synthetic organisms are limited and highly controlled. But as our tools improve, we may end up creating new forms of life that are neither natural nor entirely artificial, but something in between. And if they're alive, do they have rights, responsibilities? Can they get bored? This leads us to consider the deepest question of all. What does it mean to be alive, to be conscious, to be us? At the edge of biology and technology, we are beginning to glimpse answers, and they may be less clear than we'd hoped. As we move from decoding DNA to rewriting it, we start to treat life less like a mystery to be solved, and more like a system to be edited. This isn't just about curing diseases or making crops more resistant to fungi. It's about changing the rules biology has followed for billions of years. No pressure, obviously. In many ways, DNA is the world's longest-running operating system. And, until recently, it was read-only. Now, using tools like CRISPR, base editors, and prime editing, we can type directly into that code without breaking the whole thing in the process. That's useful when trying to remove a mutation that causes, say, sickle cell anemia. Slightly less helpful if done by someone who's had a very strong coffee and questionable training. We've already begun using this power to engineer cells that resist viruses, break down plastics, or glow in the dark, because obviously fluorescent fish were a scientific priority. But the real potential lies ahead, creating organisms with entirely new traits, or even abilities that evolution never got round to trying. At some point, we stop editing what life is and start designing what it could be. One frontier is programmable cells. Imagine cells that sense their environment and make decisions. Say, releasing insulin only when glucose levels rise. This isn't a science fiction pitch. Labs are already testing such designs. It's a bit like having cells with tiny to-do lists. If glucose high, deploy insulin. If cat nearby, initiate purring. That sort of thing. The bigger vision is to rewrite entire genomes. Some researchers are planning to redesign yeast and bacteria from the ground up, streamlining their DNA to make them more efficient or robust. There's even a proposal to recreate a human genome from scratch, not to clone people, but to learn how life assembles itself, step by step. It's part biology, part IKEA flat pack nightmare. With this level of control comes new responsibilities. Editing one gene might fix a disease. Editing many could shift what it means to be human. And if we start designing future generations, who gets to decide what counts as an improvement? Faster metabolism? Better memory? The ability to digest dairy without consequences? There are technical limits, of course. DNA is complex, and small changes can have unintended effects. Plus, ecosystems aren't predictable. Releasing modified organisms into the wild could go rather poorly. We've all seen what happens when someone says, "What's the worst that could happen?" and then introduces rabbits to Australia. This leads us to consider how we balance possibility with responsibility. We're no longer just observers of life. We're becoming its editors. And like all good editors, we need to know when to rewrite and when to leave well enough alone. By now, it's hard to ignore the idea that life isn't just made of molecules. It's made of information. DNA isn't just a chemical, it's a coded set of instructions. That's why we can copy it, edit it, even store digital data in it. Life, it turns out, is less like a soup and more like a spreadsheet with ambition. This shift in thinking has huge implications. If organisms are based on information, then biology becomes something you can program. That's not a metaphor. Scientists are now writing genetic code the way you'd write software, although with slightly higher stakes. If your phone app crashes, it doesn't usually wipe out a colony of engineered bacteria. The information view also helps explain things like heredity. You don't pass on your muscles or opinions, you pass on code. That code is read, interpreted, and turned into physical traits. It's not a flawless process. Errors creep in, but it's robust enough that jellyfish, oak trees, and accountants can all trace their ancestry back to the same informational root. This leads us to a deeper realization. Evolution itself is an information system. It generates variation, tests it against the environment, and stores the results in DNA. Like a deeply committed algorithm, it keeps running experiments, generation after generation, occasionally discovering something useful, like opposable thumbs, or the platypus. Understanding life as information also opens new frontiers. We can now simulate evolution on computers, create artificial life forms using software, or design DNA with code before even touching a lab bench. Entire companies exist solely to print custom DNA from emailed instructions. It's biology by post. This informational framing isn't just a clever analogy. It has real consequences for how we approach science. The lines between biology, computer science, and engineering are blurring. Genes become logic gates. Cells act like processors. And if you find yourself debugging a yeast genome at 3 a.m., you're probably not alone. It also forces us to ask whether life is unique to earthly chemistry, or whether any system that stores and processes information might count as alive. Could we one day find silicon-based organisms, magnetic ones, entirely virtual ones? If DNA is just one format life can use, perhaps the universe is more alive than we currently recognize. This leads us naturally to our final destination, life beyond Earth. If information is the essence of life, then it might not be limited to carbon, water, and the occasional mitochondrion. It might be hiding in places we haven't even thought to look yet. As we begin to understand life as information, the next logical question is, where else might that information be running? Earth, for all its variety, is just one planet orbiting one star. And there are hundreds of billions of stars in our galaxy alone. If life happened here, surely it's had other chances elsewhere. Unless, of course, the universe is just being awkward. The search for life beyond Earth usually starts with familiar ingredients: liquid water, carbon-based molecules, a reasonably calm environment. That's why Mars, Europa, and Enceladus keep cropping up in scientific conversations. They're nearby, potentially wet, and not immediately hostile, which, as far as space goes, is a glowing review. On Mars, we've found dried-up riverbeds and underground ice. No life yet, but the planet clearly had a wetter past. Europa and Enceladus, moons of Jupiter and Saturn, may have oceans beneath their icy crusts, kept warm by gravitational tugging. Yes, it's a bit of a stretch, but so was the idea of continental drift, and now we teach it to 12-year-olds. We also scan the skies for signals, bursts of radio waves, laser pulses, or patterns that don't look like cosmic noise. Projects like SETI search for extraterrestrial intelligence keep an ear out for alien broadcasts. So far, we've heard nothing conclusive. Either no one's calling, or they've screened our atmosphere and decided to block our number. But the most promising method might be exoplanets. We've now identified thousands of planets orbiting distant stars, some in the habitable zone, where temperatures might support liquid water. A few even seem to have atmospheres with hints of oxygen or methane, which sounds hopeful, unless you realize cows also emit methane. So, cautious optimism. The big question is, would life elsewhere look anything like ours? Carbon might be common, but alien life could be based on silicon, ammonia, or something we haven't dreamt of. It might not have cells, DNA, or anything we'd recognize as biology. We might already be staring at it through telescopes and completely missing the point. There's also the time problem. The universe is nearly 14 billion years old. Civilizations could rise, fall, and leave no trace long before we ever get a whiff of them. So even if life is common, it might still be functionally invisible, like your neighbor's Wi-Fi network. Technically there, but not something you can access or understand without the password. This leads us to a final thought. We may be rare, but we may not be alone. And even if we never find alien microbes or chatty civilizations, the act of searching expands our view. It reminds us that Earth is not the center of anything, except, perhaps, our own attention. We've traveled from molecules to minds, cells to stars, and now we find ourselves somewhere awkward, the boundary between what we know and what we don't. Science has done an impressive job explaining life's workings, but it hasn't cracked everything. And nature, as it turns out, is under no obligation to make things easy. Take consciousness. We know the brain is made of neurons, connected in complex networks, sending electrical signals. But how that gives rise to a sense of self, to the experience of being, remains one of the biggest mysteries. It's like trying to explain a concert by analyzing the wood in the violin. Technically accurate, completely unsatisfying. Then there's the origin of life. We've got plausible theories. Chemical evolution, self-replicating molecules, maybe even RNA as a kind of biological bootstrap. But no one's yet demonstrated the full path from non-living chemistry to living cells. It's not that we're clueless, just that the recipe seems to be missing a few steps, and the oven exploded several billion years ago. Other gaps are more mathematical. Evolution explains complexity without needing a designer, but predicting exactly what will evolve, or when, is fiendishly hard. Biology isn't like physics, where equations describe motion with clean precision. Living systems are messy, adaptive, and constantly rewriting the rules. It's more like jazz than clockwork. And of course, there's the big question. Why is the universe the way it is? Why these laws, these constants, this set of particles? Some physicists suggest there might be countless other universes with different settings. Ours just happens to be one where chemistry works and life is possible. A bit like finding yourself in a restaurant that serves food you can eat, instead of boiling tar. Uncertainty isn't a sign of failure, it's a feature of science. The more we learn, the more refined our questions become. We used to wonder what lightning was. Now we ask why quantum fields behave the way they do. Not necessarily easier, just more sophisticated ways of being confused. What matters is how we handle that uncertainty. Good science doesn't fill gaps with guesses. It explores, tests, and revises. Sometimes it confirms what we suspected. Sometimes it throws us completely. Either way, the frontier keeps moving, and we follow, notebook in hand, hoping the next discovery isn't hidden behind a black hole or an ethics committee. So, as we arrive at the edge of our current knowledge, one truth remains. Life, for all its complexity, is something we can study, shape, and understand, at least partly. What lies beyond? That's tomorrow's question. After all the atoms, cells, and evolutionary tangents, we return to a question so simple it's irritating. What is life, really? We've spent the last few billion years watching molecules organize, reproduce, mutate, and sometimes throw tantrums in Parliament. But is that all life is? A set of behaviors? Or is there something deeper holding it all together? One popular answer is that life is a process, not a thing. Living systems are defined by what they do: grow, adapt, reproduce, maintain internal order. Take away those activities, and what you've got left is, well, compost. But the activities themselves aren't magic. They arise from chemistry. Specifically, from networks of reactions that somehow keep going. Like a very determined chain letter. Another view says life is about information. Living things don't just react, they record, transmit, and edit instructions. DNA stores data, proteins read it, cells make decisions based on it. Life in this sense is a system for managing information in a changing world, which makes every organism a bit like a biological USB stick, slightly less portable, much harder to back up. Some scientists have suggested that life might best be defined thermodynamically, as a system that maintains order by consuming energy and increasing disorder elsewhere. That's how your body stays alive, by breathing in oxygen, eating food, and turning both into heat and waste. Life doesn't defy physics, it just plays it rather cleverly to its advantage. There's also a growing idea that life is self-organization plus memory. That is, systems that don't just form patterns, but remember and improve those patterns across time. Evolution is the memory. Natural selection is the editor. It's trial and error at planetary scale. Occasionally brilliant, frequently chaotic, always unpaid. Of course, none of these definitions are perfect. Viruses, for instance, copy themselves and evolve, but only inside other cells. Prions, a sort of rogue protein, don't even have genes, yet they spread like biological gossip. Are they alive? Scientists still argue, and as always, the more we look, the blurrier the line becomes. Perhaps the best we can say is that life is not one trait or substance, but a kind of pattern, a process that persists, adapts, and resists decay for as long as possible. It doesn't need to be made of cells. It doesn't need to resemble us. It just needs to keep going for one more moment in defiance of entropy and indifference. This leads us to our final stop. If life is a pattern, and patterns can emerge anywhere the right conditions exist, then maybe life is not rare at all. Maybe it's what matter does when left alone with enough time. A rather elegant mess, repeated across the universe. If life is a pattern that emerges under the right conditions, then we're left with one final question. How common are those conditions? Is life an astonishingly rare event, like a perfect game of darts in a thunderstorm? Or is it the kind of thing the universe keeps trying, like teenagers attempting to cook pasta without supervision? The honest answer is, we don't know. Earth is the only place where life is confirmed to exist. That makes the sample size 1, which, statistically speaking, is not ideal. Imagine trying to guess how many people like pineapple on pizza after meeting just one person. And that one person is you. Still, we can make educated guesses. Life needs a few things: stable energy, liquid water or something similar, and a platform for chemistry, usually a planet. These aren't especially rare. Planets are common. So are stars. And water turns up all over the place. From Mars' ice caps to moons like Europa and Enceladus, where liquid oceans lurk beneath frozen crusts. Then there's the chemistry. Carbon, hydrogen, oxygen, nitrogen. The basic elements of life are some of the most abundant in the universe. Organic molecules have even been found on comets and interstellar dust. In other words, the ingredients are everywhere. What we don't yet know is how often they end up stirred into something alive, rather than just left floating about, unused, like gym memberships in January. Some scientists lean towards life being inevitable, arguing that once conditions are right, chemistry naturally heads in life's direction. Others think the transition from molecules to metabolism to memory is so complicated, it might have been a fluke, a one-off, a cosmic typo that somehow worked. This is where the search for life elsewhere becomes so important. Mars, Europa, Titan. If we find even the faintest traces of life beyond Earth, it suggests that life isn't unique. It's what matter does when given time, warmth, and a bit of molecular flexibility. But if we search and find nothing, well, that would be a bit awkward. Still interesting, just more existentially lonely. The truth probably lies somewhere in between. Life may not pop up on every planet, but it may not be vanishingly rare either. Like mushrooms in the woods. It takes the right conditions, and you have to look in the right place. And ideally, not step on them. As we end our journey, we return to where we began. A single planet, orbiting an unremarkable star, filled with the only confirmed life in the universe. For now, whether we're alone or surrounded by unseen neighbors, one thing's clear: we are part of something vast, unfolding, and still mostly unknown. We've explored atoms, stars, cells, death, memory, slime molds, and the odd sea cucumber, all to understand what life is. Now, inevitably, comes the question some people like to save for late-night chats or philosophy tutorials. What's the meaning of it all? Why does life exist at all? And more to the point, why does it keep getting up in the morning? From a scientific standpoint, life doesn't need a reason. It just is. Evolution doesn't have goals. Molecules don't aspire to greatness. Life exists because chemistry allows it. And it continues because some arrangements of matter are better at lasting than others. It's not glamorous, but it's impressively efficient. Like a stubborn stain that won't go away. But meaning is a different beast. Unlike genes or proteins, it's not something you can isolate in a lab. Meaning is what minds assign to patterns, and conveniently, minds are something life invented. Humans, with our oversized brains and unnecessary existential crises, are especially good at asking why, when the universe only offers because. Many people find meaning in connection. Family, friends, shared experiences. Others find it in progress, in discovery, in solving maths problems no one else cared about. Some just enjoy snacks. From a biological point of view, this is all perfectly normal. Our minds evolved to look for patterns and stories. It's how we make sense of things. Even if sometimes the pattern is, this hurts less when I have a biscuit. Interestingly, science doesn't strip life of meaning. It just doesn't provide it directly. What it does offer is perspective. The knowledge that you, me, and every mushroom are made of atoms forged in stars. That we're part of a process stretching back billions of years, and possibly repeated on other planets. It's not a purpose, but it is a place in the story. Some thinkers suggest that meaning comes from action. That is, life gains value by what it does, by building, helping, creating, or simply enduring. From this view, meaning isn't handed to you. It's assembled slowly, like flat-pack furniture. Often backwards, occasionally with missing screws. So perhaps life doesn't need a singular meaning. Perhaps it's enough that life exists against long odds and keeps going. That it can ask questions like this, and occasionally laugh at itself along the way. A weird little bubble of order, floating in an indifferent universe, wondering what it's for. And with that, we end not with a neat answer, but with the right kind of question. What does it mean to be alive, here, now? Only you can answer that. Preferably after a good sleep, a decent sandwich, and, if you must, some pondering beneath the stars. All life is local. That is to say, it exists in specific places, under specific conditions. Earth's surface is one of those rare environments where liquid water, breathable air, and moderate temperatures overlap. It's like a habitable Goldilocks zone, except, instead of porridge, there's just nitrogen, oxygen, and quite a lot of mud. But even on Earth, life is fragile. We're used to seeing it everywhere. Trees, insects, that moss growing on your bathroom tiles. But ecosystems are built on tight balances. A few degrees warmer, a few chemicals out of place, and things unravel. Past mass extinctions have shown how easily life's intricate networks can collapse. At the heart of this fragility is dependence. Life depends on stable climate, clean water, fertile soil, and each other. Disrupt one part. Burn enough fossil fuel? Remove too many forests? Acidify too much ocean? And entire systems can shift or fail. This isn't hypothetical, it's happening. Coral reefs are bleaching. Ice caps are melting. And some scientists now track insect populations with the kind of anxiety usually reserved for exam results. This isn't to say Earth will become uninhabitable overnight, but it is changing. And while life in general might adapt, evolution is persistent like that. Human life as we know it depends on very specific conditions. Cities, agriculture, even sleep schedules, they all expect a stable world. If we tip things too far, we may find ourselves perfectly adapted to a planet that no longer exists. On longer timescales, there's a deeper issue. Our sun is slowly getting hotter. In a billion years or so, Earth's surface will become too warm for liquid water. Life may retreat underground or into the oceans, but the clock is ticking. Eventually, even this unusually lucky planet will run out of time. The universe giveth, and eventually, the universe roasteth. Which raises a quiet question: is life destined to burn out, or could it spread? We've developed technology capable of sending machines, and possibly, one day life itself, beyond Earth. The idea isn't science fiction, it's contingency planning. If life is rare, then preserving it might be one of the most important things we ever do. Not because we're special, but because life is. We often speak of the universe as cold, vast, and indifferent. And mostly, it is. But here, on this spinning rock, there's a tiny patch where molecules became aware, where carbon learned to worry. Life is not the default state of matter, it's an exception. And like all exceptions, it needs looking after. This leads us to consider one last possibility, that in a universe where life is rare, the job of preserving it may fall to life itself, not through grand heroics, but through the quiet act of care for each other, and for the only home we've ever known. And so, from atoms to animals, microbes to minds, we arrive at something strange. Not just life, but life that asks questions, that builds microscopes and telescopes, that wonders where it came from and what it's doing here. In other words, us. Quite an awkward species, but the only one we know that's actively trying to understand the entire universe, whilst also arguing about pineapple on pizza. Self-awareness is not a biological requirement. Amoebas manage just fine without existential crises. Yet humans, and perhaps some other animals, possess this added layer, the ability to reflect. We can imagine the past, the future, and alternative versions of both, where we remembered to do the washing up. What makes this interesting is scale. We are made of the universe. The iron in our blood was forged in stars. The oxygen we breathe comes from tiny bacteria and plants. Our thoughts emerge from electric signals in jelly-like brains. We are, in every literal sense, part of the cosmos. And yet we observe it, study it, try to explain it. That's new. Carl Sagan once described humans as a way for the cosmos to know itself. Which is poetic, but not entirely wrong. Consciousness, particularly the kind that builds radio telescopes and throws tantrums on Twitter, allows the universe to be noticed. Not worshipped, not designed, just noticed. A process of atoms, becoming aware of atoms, talking about atoms. Slightly absurd, slightly profound. Of course, none of this means we're central to the story. Earth is one planet, orbiting one star, in one galaxy, among billions. Intelligence may well exist elsewhere, though we haven't seen so much as a polite wave. But it does mean we're in the story, part of the system, active participants in the grand experiment. This also comes with responsibility. We're clever enough to split atoms, rewire genes, and overfish the oceans, which means our decisions matter. Not just to us, but potentially to the future of life itself. It's a strange twist of evolution. The species that understands extinction is also the one most likely to cause or prevent it. So here we are, self-aware, socially awkward, and semi-permanent. A species capable of both poetry and plastic waste. And yet, through science, we've managed to glimpse the history of the universe, the machinery of life, and the fragility of our future. Not bad, considering our closest relatives throw their own poo. This leads us to one final reflection. If the universe can evolve beings that question their own existence, perhaps its story isn't just about stars and space. Perhaps it's also about curiosity. Not because the universe needs to be understood, but because, somewhere in it, life arose that simply could. After all that, it's probably time for a nap. We've traveled from the birth of atoms to the question of meaning, wandered through cells and stars, language and memory, fungi and feelings. Now we land gently, back in the here and now, on a small planet, under a cooling sky, where life goes on whether or not you've figured it all out. Science doesn't always provide comfort. It's not designed to. But there is something quietly reassuring about understanding how things work, or at least how they might. No magic required. Just atoms, energy, and a set of rules that occasionally allow molecules to ponder themselves, usually while procrastinating on a Monday. What we've seen is that life is a process, not a product. It changes, adapts, occasionally loses its keys, and what defines it isn't perfection, but persistence. Life tries things, fails a lot. Tries again. Not very glamorous, but surprisingly relatable, especially if you've ever attempted IKEA furniture without instructions. And yes, there's plenty we still don't know. Consciousness remains partly mysterious. We've no idea how common life is in the universe. And to be honest, we're still arguing about the best way to define a species. But that's part of the point. Uncertainty isn't a flaw. It's the reason science keeps going, like a very polite argument with the universe. Meanwhile, the fact that life exists at all, and that it's capable of laughing at itself, is rather remarkable. Not miraculous, just rare, and worth looking after. Because whether it's in a rainforest, a coral reef, or the back of your fridge, life represents billions of years of chemistry that somehow didn't give up. So perhaps the best thing we can do with our brief conscious window is to stay curious, to ask good questions, to look after each other and the planet we currently call home, and maybe, just maybe, to rest now and then. Not everything needs solving before bedtime. After all, you're part of this long, improbable story, one link in a chain stretching from stardust to neural networks. And whether you're feeling inspired, puzzled, or just ready for a biscuit, that's all right. You're alive, you're aware, and you made it this far. So sleep well. None of this happened by accident. Well, the universe might have, but the work of stitching it together into something you could fall asleep to definitely didn't. So before we vanish into the metaphorical darkness, a few acknowledgements. Like any good experiment, this one involved a team. Some wrote, some checked. Some very patiently corrected the spelling of anaerobic several times. The science we've explored tonight builds on thousands of studies, decades of research, and quite a lot of academic debates held over weak coffee. From Darwin's finches to the Large Hadron Collider, none of these insights came from one place. They came from people across cultures, languages, and lifetimes, all asking their own version of why. Much of the biology here is based on decades of work in evolutionary theory, cell science, genetics, ecology, and cognitive science. We've drawn heavily on published consensus in journals, open access reviews, and textbooks. Whenever there was uncertainty or debate, we've tried to say so. Unlike your uncle on Facebook, science is usually quite happy to admit it doesn't know everything. The narrative arc. The decision to start with chemistry and end with consciousness owes a quiet debt to many educators and communicators. Carl Sagan, Richard Feynman, Mary Roach, and Sabine Hossenfelder, to name just a few. If you ever want to go deeper, their books are a good place to start. Though maybe don't bring one to bed, unless you enjoy falling asleep mid-sentence. A note on language. We've used British spelling throughout, and kept explanations clear and grounded. If anything felt oversimplified, that was intentional. Complexity can always be added later. But clarity takes work. And a fair bit of deleting. Our thanks also go to the many unsung contributors. The scientists whose datasets, experiments, fieldwork, and late nights made this understanding possible. And the teachers who managed to explain meiosis without sighing loudly every 5 minutes. True heroes. And finally, to you, for your curiosity, for choosing to spend a bit of your short life learning how life itself came to be. There are worse ways to spend your time, though watching paint dry does come close, scientifically speaking. Now, that's it. You're all caught up. The story of life, the universe, and quite a bit in between. Sleep well. Dream safely. Try not to think about quantum mechanics. If you're still awake, well done. Either this was more interesting than expected, or you've got a very patient bladder. Before we go, a few footnotes. Not the tiny font kind found in dusty textbooks, but small clarifications and reminders, just in case anything lingered in the back of your mind, like a forgotten biscuit in a coat pocket. Firstly, the science in all of this is current as of the time it was written, which is to say, right now, it's accurate. But science doesn't sit still. It updates. It revises. Occasionally it throws everything out and starts again, which is comforting if you've ever had to redo a spreadsheet from scratch. So if something here sounds off 10 years from now, that's probably a good sign. It means we've learned more. Secondly, models are just that. Models. We use them to represent how things might work, not to claim that we've nailed it. A cell diagram isn't what a real cell looks like. A tree of life isn't drawn to scale. And a diagram of the Big Bang isn't going to come with surround sound or lava effects. They're helpful. They're not perfect. Bit like us, really. A quick word on uncertainty. Science deals with probability, not absolute truth. Terms like likely, suggests, or appears to be aren't signs of weakness. They're signs someone's doing their job properly. If you hear a scientist sounding very confident about everything, they're either joking or selling something. Now on terminology. We've tried to use plain English throughout. But even plain English can be a bit slippery. Words like life, species, and intelligence have working definitions, but they're not fixed in stone. You'll find biologists, philosophers, and chatty pub patrons all using the same word in quite different ways. Sources? This project builds on a wide range of publicly available research, peer-reviewed journals, academic reviews, textbooks, and large-scale science communication efforts. In particular, material has been checked against current biological, astronomical, and cognitive science consensus. If you're the type who enjoys chasing references at 2 a.m., you are not alone. Finally, nothing here is meant to give you all the answers. Quite the opposite. If anything, we hope it's left you with better questions. Like, why does life bother? How much can we ever know? And why do mushrooms seem to be involved in absolutely everything? We're still looking into that one. And that brings our journey to a gentle close. If you're still with us, you're welcome to drift off with another of our sleepy deep dives. Until then, rest well, and good night.